So good afternoon everybody. Today we are going to discuss about the molecular basis of inheritance. Molecular basis of inheritance.
Good evening. Okay, start answer sir, please. మీరందరూ రెడీగా ఉన్నామండి డౌట్లు అడగమని చెప్పండి.
జీబీ సార్
Basis of inheritance.
ఒక నంబర్ ఇస్తారు స్టూడెంట్ దానికి అడగమని చెప్పండి.
So according to Mendel, we know the pattern of inheritance. According to Mendel, we know the pattern of inheritance. Why is not there?
So the students who are listening the class, so please note down if you have any doubts or any queries, please note it down in your notebooks, and at the ending of the class we will giving you 15 to 10 minutes of time, so you can ask the questions through the phone number we are providing on the screen. So all the students should note down your doubts. So whenever the సార్ క్లాస్ చెప్తున్నప్పుడు ఎవరికి ఎటువంటి సందేహాలు డౌట్లు వచ్చినా కానీ క్లాస్ ఎండ్ అప్పుడు మీకు ఒక నంబర్ ఇస్తాము. ఇప్పుడు మీరు చూస్తున్న యూట్యూబ్ లైవ్లో ఒక నంబర్ ఇస్తాము. ఆ నంబర్కి కాల్ చేసి సారు డౌట్లు అడగండి, సార్ మీ డౌట్స్ క్లారిఫై చేస్తాం.
So, okay. According to Mendel, we know the pattern of inheritance and the genetic basis of inheritance. Genetic basis of inheritance. But Mendel mentioned the factors, but the chemical nature of factors, that regulate the pattern of inheritance. Was not clear, was not mentioned, was not clear. So Mendel mentioned the factors, but he did not provide any proof for the existence of factors. That means the chemical. What is the chemical of the factors that regulate the pattern of inheritance? Right. Over the 100 of years, it was proved that the chemical nature of inheritance is that DNA. It was proved that the chemical nature of inheritance, that is the DNA. So DNA is the chemical nature of inheritance. DNA is the chemical nature of inheritance. But Mendel did not mention the DNA. Only the factors are there. They regulate the pattern of inheritance. After Mendel, over the 100 of years, the chemical nature of DNA was identified by many experiments. So DNA is the molecular basis of inheritance. What is the molecule of inheritance? What is the molecular basis of basis of inheritance. So that is the question. So the answer is DNA. So DNA is the molecule that is the basis of inheritance. Where is the location of DNA? Where is the DNA is located? That is nothing but chromosome. The location of DNA is chromosomes. Chromosomes, right. Location of DNA is chromosomes. So what is present in chromosomes? What is present in chromosomes? So right. So it is the chromosome. Right, it is the DNA. It is also chromosome. It is the DNA. Right. So these are the chromosomes. One is single chromatid chromosome. Single chromatid. Single chromatid, two chromatids. So both are chromosomes, single chromatid chromosome and two chromatids present in the chromosome. That is nothing but single DNA. Single DNA is there and two DNAs are there. Two DNAs are there. Nothing but what is chromatid? Chromatid is equal to chromatid, nothing but chromatid is equal to DNA. Chromatid nothing but DNA. So DNA is the chromatid. Single chromatid nothing but it is present before the S phase of cell cycle. Before the S phase, we see the single chromatid chromosomes, and after the S phase of cell division, we see the chromosome with two chromatids. Two chromatids before S phase and after S phase. Single chromatid chromosome and two chromatid chromosomes, nothing but two DNAs. That means replication. Replication doubles the DNA. Replication doubles the DNA that takes place in S phase. Replication doubles the DNA that takes place in S phase, right? Doubles the DNA. So it takes place in S phase. S phase of cell cycle. S phase of cell cycle, right? So that is the DNA. But what is present in the DNA? Nothing but, so these are all the segments. So segment of DNA is called gene. Gene nothing but DNA, but a part of DNA, a segment of DNA. Segment of DNA is called gene. Segment of DNA is called gene. So it is the generally basis. Gene is the basis. Gene is expressed to protein. So gene is genetic makeup is the gene that is expressed to form phenotypes. We can see the phenotype, that is the physical appearance of an organism, that is regulated by the genes present in the chromosome. Nothing but a segment of DNA is called a gene. So these are all genes. These are all genes, right? So no, totally the DNA is the genetic material proved by several experiments. Right. So DNA is the genetic material. DNA is the genetic material in majority of organisms. Majority of organisms. RNA also act as genetic material in some viruses. Now, what is the duty of RNA? RNA act as genetic material in some viruses, as well the second function, messenger. For example, mRNA. So mRNA act as the messenger RNA. So second, third one, adapter molecule. Adapter, nothing but tRNA. So messenger RNA carry the genetic information from DNA to protein, that is the messenger RNA, and adapter RNA, tRNA, that carry the amino acids from cytoplasm to the ribosomes for the construction of a protein. Right, the fourth one, structural RNA. Structural, nothing but rRNA. rRNA act as structural RNA. Next to catalytic RNA. So which are called generally ribozymes. Ribozymes, for example, 23S or RNA. So it is catalytic RNA like enzymes. So it increases the rate of reaction, right? Catalytic RNA. So these are the roles. So first one is genetic material in some viruses, as well the second one, messenger. Messenger, nothing but it carries the genetic information from DNA to proteins, right? Then adapter molecule, it carries the amino acids from cytoplasm to ribosomes for the construction of a protein, and the structural RNA. So it is present in the ribosomes. So ribosomes are constructed by rRNA, so they are called structural RNAs, right? Then catalytic RNA. So 23 srRNA act as a catalytic, nothing but ribozymes antam. RNA act as enzyme, that is called ribozyme. RNA act as enzyme, that is called ribozyme. So initially DNA is the genetic material in majority of organisms. It was proved by several experiments. But as well, RNA also act as genetic material in some viruses. But RNA act as several activities, several functions RNA involved. Those are messenger, adapter, and structural and catalytic roles. Catalytic roles, right? Now, where the location of DNA and RNA? DNA, RNAs are present in the nucleus in prokaryotes. So DNA as well RNA are present nucleus of eukaryotes. Right. Now, what are DNA, RNA? So, which are biomacromolecules. Biomacromolecules. Nothing but biomolecules, especially macromolecules. So, which are polymers. Polymers. So, DNA and as well RNA both are polymers. Polymers means which are made by several monomers. What are the units of polymer? DNA as well RNA. So, those are the nucleotides. Nucleotides are the monomers of DNA as well RNA. Nucleotides, which are the building blocks of nucleic acid. So, DNA, RNA present in the nucleus of eukaryotes, which are nucleic acids. Nucleic acids. Nucleic acids are polymers. Proteins are polymers. As well, polysaccharides are polymers. You know, polysaccharides. Proteins, nucleic acids. So, which are generally polymers, which are made by several monomers. Now, DNA is DNA as well RNA are heteropolymers. Heteropolymers means made by different kinds of monomers. Made by different kinds of monomers. Nothing but different types of, there are different types of nucleotides. Different types of nucleotides are there, which form the DNA as well RNA. Right. Who discovered the nucleic acids initially? So, Frederick Meischer. Frederick Meischer in 1869 identified nucleic acids. So, and named it as nuclein. So, nuclein. So, initially Frederick Meischer is the scientist. In 1869, he first identified the DNA in the nucleus and named it as nuclein. So, after that, in 19 James Watson and Francis Crick. James Watson and Francis Crick revealed the structure of and discovered the structure of DNA. Structure of DNA in 1953. Revealed the structure of DNA in 1953. Nothing but a double helical structure proposed by Watson Crick. Double helical structure proposed by Watson Crick in the year 1953. based on, what is the base of Watson and Crick proposed double helical structure? What is the base? So one is Wilkins Franklin, Wilkins and Franklin X-ray diffraction method. X-ray diffraction method or technique. Second one, Erwin Chargaff, chemical analysis. So Erwin Chargaff, chemical analysis. Then the third one, Pauling hydrogen bonds. So these are the three scientists. Based on their experiments, Watson and Crick proposed the double helical structure of DNA. So one of the bases Wilkins Franklin X-ray diffraction method, as well Erwin Chargaff chemical analysis. Chargaff rule. Chargaff proposed a rule that is called Chargaff rule. According to this, purines and pyrimidines are equal. Pyrimidines are equal. Nothing but 1:1. Purine, adenine is equimolar with thymine. Guanine is equimolar with cytosine. Adenine equimolar with thymine, guanine equimolar with cytosine. So A plus Z is equal to C plus T. A plus Z is equal to C plus T, or A plus G by C plus T is equal to 1. So it is the Chargaff rule. Erwin Chargaff proposed the Chargaff rule. According to this, purines and pyrimidines are equal in the structure of DNA. Purines and pyrimidines are equal in the structure of DNA, that is 1:1. Adenine is equimolar with thymine, as well guanine is equimolar with cytosine. A plus Z is equal to C plus T. A plus Z by C plus T is equal to 1. So it is the Chargaff rule. So based on this experiment, Mendel proposed the structure of DNA, as well Pauling hydrogen bonds. Pauling hydrogen bonds helps the discovery of the structure of DNA. Right. So the structure of DNA. So it is the double helical structure. So totally 2 strands. DNA is made up of two polynucleotide chains. So this is the one strand, this is the another strand. There are 2 strands which make the DNA. So DNA is made of 2 polynucleotide chains. Sugar phosphate is the phosphate is the backbone. Sugar phosphate is the backbone, right? So for example, right, sugar phosphate is the backbone. So two strands are anti-parallel. Two strands of DNA. Two strands of DNA arranged anti-parallel. Arranged anti-parallel manner. So for example, if one strand polarity is 5 prime to 3 prime, the other strand polarity is automatically 3 prime to 5 prime. One strand has the polarity 5 prime to 3 prime. The opposite strand has another strand has the polarity 3 prime to 5 prime. Next, third one. So DNA is made up of two polynucleotide chains. Sugar phosphate is the backbone as well. The nitrogen bases, N bases, nitrogen bases project inside. So right. So this is totally an helic. Helic nothing but 360-degree coiling. Two strands right-handedly, two strands of DNA, coil right-handedly to form helix. Nothing but 360 degrees turn. 360 degrees turn is called a helic. And helic, so the 360-degree turn in which generally 10 base pairs are there. 10 base pairs are present in a helic. So this is totally the helic, nothing but having 10 base pairs, right? So in which always purine binds with pyrimidine. Always, right, purine binds with pyrimidine by weak hydrogen bonds. Always purine binds with pyrimidine. So for example, adenine binds with thymine by 2 hydrogen bonds, and guanine binds with cytosine by 3 hydrogen bonds. Adenine binds with thymine by 2 hydrogen bonds, guanine binds with cytosine by 3 hydrogen bonds. So which are generally weak hydrogen bonds, right? In which 10 base pairs. So this is the structure of DNA in which 10 base pairs are there. Base pairs are there. Next, fifth point, the plane of arrangement of one base pair stacks over the other in a helix. The plane of arrangement of one base pair stacks over the other in a helix along with hydrogen bonds provide the additional stability to DNA. Additional stability to DNA. The plane of arrangement of one base pair stacks over the other in a helix along with hydrogen bonds provide the stability to DNA. So right. So DNA is made, right. For example, 5 prime, 3 prime, 5 prime, 3 prime. So this is the helix we know, right, in which 10 base pairs are there. The helix angle is angle of helix is 360 degrees. The distance of the pitch, helix nothing but pitch, form a pitch, 360 degrees turn, form a pitch. The distance of pitch is 3.4 nanometers, otherwise 34 angstroms. Helix angle is 360 degrees. In between two successive base pairs, between two base pairs, the angle is 36 degrees. Distance between helix is 34 angstroms. Between two base pairs, the distance is 0.34 nanometers, otherwise 3.4 angstroms. So it is the distance between the helix. Distance of helix is 34 angstroms, otherwise 3.4 nanometers. There are 10 base pairs. So, in between two base pairs, the distance is 3.4 angstroms, otherwise 0.34 nanometers. 3.4 angstroms are otherwise 0.34 nanometers, but the helix distance is 34 angstroms, otherwise 3.4 nanometers. The width of the helix, width of the DNA, nothing but the width of the DNA is 20 angstroms. So it is constant. Width of the DNA, width of DNA is 20 angstroms. So it is common for entire DNA. It is common for entire. Total DNA show the same width. That is because of why the DNA show the same width in between two strands entirely. That is because of always purine binds with pyrimidine. Purine binds with pyrimidine. Always purine binds with pyrimidine that maintain the 20 angstroms width of DNA. It is constant entire DNA. The constants is because of always a purine binds with pyrimidine. A purine binds with pyrimidine. Right. Purine binds with pyrimidine. So that maintains the constant distance in between two strands of DNA entire because of one purine binds with one pyrimidine. What is the reason behind that? For example, purine has two rings, pyrimidine has single ring. Purine has two rings, pyrimidine has single rings. As well, so it is the pyrimidine, so it is the purine, it is the purine, so it is the pyrimidine. So always three rings are present in between two strands. Always any base pair constitute of three rings. Every base pair, this is totally called base pair. One purine, one pyrimidine binded by hydrogen bonds, they form a base pair. Base pair nothing but two nucleotides. Two nucleotides are united by the hydrogen bonds. So each base pair consists of a purine and a pyrimidine. Always purine has two rings, pyrimidine has single ring. So always every base pair show the three rings. That is why the distance between two strands of DNA is entirely similar. Entirely similar. Right. What is the structure of DNA? So DNA, already we know DNA is polynucleotide chains. How many chains? And two chains. Polynucleotide chains. DNA is made up of polynucleotide chains. So, what is the building block of polynucleotide? Nothing but a nucleotide. Nucleotide is the building block. Building block of DNA as well RNA. Nucleotide is the building block of DNA as well RNA. So nucleotide is made up of a nitrogen base plus sugar, pentose sugar, pentose sugar plus phosphoric acid. Nitrogen base, pentose sugar, phosphoric acid. So nitrogen base. What is the structural components of nucleotide? Nucleotide is made up of three components. One is nitrogen base, another one is pentose sugar, and the third one is phosphoric acid. Nitrogen bases. So which are two types of nitrogen bases are there? two types of nitrogen bases. So one is purines, so which are generally heterocyclic and bicyclic. Heterocyclic bicyclic rings. Heterocyclic bicyclic rings in which 1, 3, 7 and 9th position are nitrogens. 1, 3, 7, 9 positions are nitrogens. Right. Purines are made up of, generally purines are heterocyclic and bicyclic rings in which 1, 3, 7, 9 positions are nitrogens. Purines are of two types. One is adenine. So right. Second one is guanine. Adenine and guanine. These are the two purines. So we see the structure of adenine. Adenine, also called 6-aminopurine. 6-aminopurine. So right, adenine is 6-aminopurine. The chemical structure is C5, H5, N5. Right, it is double ringed nitrogen base. Double ringed nitrogen base. There are two rings. So in which 1, 3, 7, 9 positions are nitrogens. So it is the nitrogen and carbon and nitrogen, carbon, carbon. So carbon, nitrogen, carbon, nitrogen, carbon. So it is the skeleton of purine. It is the skeleton of purine in which 1, 3, 7, 9. So these are the nitrogen positions. 6-aminopurine. 6-aminopurine. So it is the sixth, it is the second, fourth, fifth, sixth, seventh, eighth, ninth position. Totally, it is heterocyclic ring. Heterocyclic means the adenine is made up of not only complete carbons, it is made with carbon and nitrogen, so that is heterocyclic. Here carbons are there, nitrogens are there. That is, for example, benzene is homocyclic. That is nothing but benzene is totally made by carbons. So that is homocyclic. Here it is heterocyclic. Here carbons and nitrogens are there. So 6-aminopurine. It is the sixth carbon, amino. So purine. It is totally purine. Purine means bicyclic heterocyclic ring. 1, 3, 7, 9 positions are nitrogens. Right. 6-aminopurine. 6-aminopurine. Right. So right, C5, totally carbon numbers 1, 2, 3, 4, 5, C5, H5, 1, 2, 3, 2, 5, H5, N5, 1, 2, 3, 4 and 5. C5, H5, N5. It is the adenine. Adenine is one of the purine. Purine is because of two rings are there. That is dicyclic as well heterocyclic rings. The second one is right to B, guanine, also called 6-oxy 2-amino 6-oxy 2-amino purine. So the skeleton is same. Nitrogen, carbon, nitrogen, carbon, 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 right. So nitrogen, carbon, nitrogen, carbon. Skeleton is same. Here 6-oxy, sixth carbon oxygen is there, and on second carbon amino group is there. 6-oxy to amino purine, right. So right, it is the guanine. It is a purine because of two rings are there, heterocyclic rings are there, nitrogen positions are 1, 3, 7, 9. 1, 3, 7, 9. 1, 3, 7, 9th positions are nitrogens. Right, guanine 6-oxy to amino purine. Carbon numbers 1, 2, 3, 4, 5. C5. Hydrogens 1, 2, 3 to 5. C5, H5. N 1, 2, 3, 4 and 5. N5 and O is there. Oxygen is there, right? That is the structure of guanine. So these two are adenine, guanine are purines. What is the purine? Purine nothing but bicyclic, heterocyclic rings, in which first carbon, third carbon, seventh carbon, ninth carbons are hydrogens, right? Adenine and guanines are purines which have two rings. Purines are always have two rings. Earlier we say that every base pair formed by one purine plus one pyrimidine. Purine plus pyrimidine constitute a base pair. Purine and pyrimidine form the base pair. Purine has two rings as well. Pyrimidine one ring, nothing but three rings present in a base pair. Three rings present in a base pair. That is why the entire width of DNA is constant, that is 20 angstroms. That is 20 angstroms, right? Next, pyrimidines. Pyrimidines which are heterocyclic and monocyclic. Heterocyclic, monocyclic compounds. Pyrimidines are heterocyclic as well monocyclic compounds in which 1, 3rd positions are nitrogens. First and third positions are nitrogens, right? Pyrimidine. So which are of three types. One is thymine and the second one is cytosine. So thymine is present only in DNA, and the third one is uracil, only present in RNA. Only present in RNA. Thymine, cytosine and uracil. Thymine, cytosine and uracil. These are the three pyrimidines. What is the structure of pyrimidines? Skeleton structure of pyrimidines? Always heterocyclic as well monocyclic compounds and single ringed compounds. One purine bind with one pyrimidine. Purine is always bicyclic. Pyrimidine is always monocyclic. Totally every base pair nothing but two nucleotides paired by hydrogen bonds. Paired by hydrogen bonds constitute a base pair. Each base pair constitute of two nucleotides. One nucleotide with the purine, another nucleotide with the pyrimidine. So always 3 rings are present at a base pair which maintain the width of DNA is 20 angstroms entirely. Right. Next, the structure of thymine. So thymine, what is the skeleton? 1379 positions are nitrogens, right? Nitrogen, carbon, nitrogen, carbon, 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 as well nitrogens. So in which first position is nitrogen and the third position is nitrogen. Remaining second position, fourth position, fifth position, as well sixth position. So it is the skeleton of pyrimidine. What is thymine? 2,4-dioxy, 2,4-dioxy, 5-methyl uracil, 5-methyl, right? 5-methyl pyrimidine, 5-methyl pyrimidine, 2,4-dioxy, 5-methyl pyrimidine, nothing but thymine, also called 5-methyl 5-methyl uracil. Thymine also called 5-methyl uracil, right? 5-methyl uracil, 2,4-dioxy. Second carbon is there, one oxygen is there, and fourth carbon, oxygen is there, 2,4-dioxy, and fifth carbon, methyl group is there, 2,4-dioxy, 5-methyl uracil, 5-methyl pyrimidine, otherwise 5-methyl uracil, right? It is the structure, right? Double bond, right? Hydrogen as well, hydrogen. So what is the chemical structure of thymine? How many C's are there? 1, 2, 3, 4, C5, carbon number 5, 1, 2, 3, 4, as well 5, C5, H, 3, 4, 5. So write C5H6 N2 O2. So it is the structure of thymine. Thymine is pyrimidine, nothing but single ringed heterocyclic ring is there. Single ring as well heterocyclic ring is there. Nothing but carbon, nitrogens are there in the ring. That is why heterocyclic, it is single ring. That refers to pyrimidine. It is the skeleton of pyrimidine. Here thymine is there. Thymine nothing but 2,4-dioxy 5-methyl pyrimidine, otherwise called 5-methyl uracil. The chemical structure is C5H6N2O2. Right. The second one is cytosine. Cytosine. Right. Cytosine. What is the base structure? Skeletal structure is nitrogen, carbon, nitrogen, carbon, 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 as well nitrogen. It is the skeleton of pyrimidine, nothing but single ring is there. So cytosine, 2-oxy, 4-amino, 2-oxy 4-amino, pyrimidine, 2-oxy 4-amino pyrimidine, right? It is the first carbon, first nitrogen, second carbon, third nitrogen, fourth carbon, fifth carbon, as well sixth carbon, 2-oxy, nothing but it is oxygen is there, 4 amino, NH2 is there, nothing but it is cytosine, nothing but pyrimidine. So double bond is there, hydrogen, hydrogen. So write carbon numbers 1, 2, 3, 4, C4, hydrogens 1, 2, 2 to 4, C4H4, N3, 1, 2, 3, O, NH2, right, CH, CH, N, CO, N, right, C4H5, C4H5, N3O, that is the chemical structure of cytosine. C4H5, N3O, it is also called 2-oxy-4-amino pyrimidine. 2-oxy-4-amino pyrimidine. Then the last one, right, uracil. Uracil, right, also called 2,4-dioxy pyrimidine. 2,4-dioxy pyrimidine. What is the skeleton structure? Nitrogen, carbon, nitrogen, carbon, 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 nitrogen. So it is the skeleton structure. First position is nitrogen, second carbon, third nitrogen, fourth carbon, fifth carbon as well sixth carbon. 2,4-dioxy, 2-oxy and 4-oxy pyrimidine, right, hydrogen, 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 so hydrogen. So what is the chemical structure? Carbon 1, 2, 3, 4, C4, H1, 2, 3, 4, H4, N2, O2. So it is the chemical structure of uracil. So these are the totally nitrogen bases. One is purines, another one is pyrimidines. Purines are dicyclic heterocyclic rings and pyrimidines are monocyclic and heterocyclic rings. Both are heterocyclic, but purines are bicycle and pyrimidines are monocyclic. So these are the nitrogen bases. So uracil is only present in RNA, thymine only present in DNA. Thymine only present in DNA. So write. Next one, sugar. Sugar, it is called ribose sugar. Ribose sugar. Second component of nucleotide. Nucleotide is the building block of nucleic acid, whether it is DNA or RNA. It is second component is sugar. It is ribose sugar. Ribose sugar, pentose sugar, right? Pentose sugar. Pentose sugar. It is of two types. A, ribose sugar, and B, deoxy ribose sugar. Ribose sugar, deoxy ribose sugar. So what is the structure of ribose sugar? Ribose sugar nothing but present in RNA, deoxyribose sugar present in DNA, deoxyribose sugar present in DNA, ribose sugar present in RNA, both are pentose sugars. What is the structure of ribose sugar? CH2OH. So what is the carbon positions? So it is the first carbon, it is second carbon, it is third carbon, it is fourth carbon, as well fifth carbon. First carbon, second carbon, third carbon, fourth carbon, fifth carbon. These are the 5 carbons are there. So the formula is C5H 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. C5H10 C5H10 O5 1, 2, 3, 4, 5. C5H10O5. It is the ribose sugar present in RNA. Here on second carbon OH is there. It is the reactive group. Reactive group present in ribose sugar. That is why RNA is more reactive. RNA is more reactive because of reactive group. Additional reactive group is there. That is present at second carbon. That is present at second carbon. It is the OH is the additional reactive group present in RNA. That is why RNA is more reactive, less stable. RNA is more reactive and less stable, right? So next, deoxyribose sugar. So what is the structure of deoxyribose sugar? OH, HOH, right, H, CH2OH. So it is the structure, right? First carbon, we know, second carbon, third carbon, fourth carbon, fifth carbon is there. On second carbon, there is no oxygen. That is why deoxy. D means, what is D? Ante removal of oxygen, right? Removal of oxygen at second carbon. Removal of oxygen at second carbon. So that is deoxy. D nothing but removal. Removal of oxygen at second carbon is deoxy. Totally it is ribose sugar. Deoxyribose sugar. Deoxyribose sugar. In RNA only ribose sugar is there. On second carbon there is additional oxygen, nothing but OH. That is the reactive group. Ikkada no OH is there, only H is there. So that is why deoxyribose sugar makes the DNA more stable and less reactive. It is the sugar, pentose sugar present in DNA, right? Next one, the third component of DNA, that is phosphoric acid. Phosphoric acid. So what is the chemical structure? H3PO4. H3PO4. So it is the phosphoric acid. So it which provide acidic nature to the DNA as well as RNA. So they are called nucleic acids. The acids present in the nucleus are called nucleic acids. The acidic nature is because of phosphoric acid, H3PO4. So right. Totally these three components form the nucleotide. Three components form the nucleotide, right? Nucleotides. Totally how many nucleotides are present? So 5 types of nucleotides are present. 5 types of nucleotides. So first one is five types of nucleotides are there, in which DNA consists of 4 types and RNA consists of 4 types. 4 types. Right. Nucleotide means nitrogen base plus pentose sugar plus phosphoric acid. Nitrogen base, pentose sugar, phosphoric acid. So nitrogen base and pentose sugar constitute the nucleoside. This totally constitute the nucleotide. Nucleotide nothing but nitrogen base, pentose sugar and phosphoric acid. It is totally nucleotide. Only nitrogen base and pentose sugar constitute nucleoside. Nucleoside constitute of nitrogen base and pentose sugar. Here the what bond is present in between these two? That is called N-glycosidic bond. N-glycosidic bond. N-glycosidic bond is present in between nitrogen base and pentose sugar. As well, the pentose sugar and phosphoric acid, there is a bond that is called ester bond. Ester bond. Ester bond is there. So phosphoric acid is the third component of DNA as well RNA, third component of nucleotide. Right, nucleotides are 5 types, in which 4 types are present in DNA, as well 4 types are present in RNA. Right, nitrogen base, pentose sugar and phosphoric acid totally constitute a nucleotide, in which nitrogen base, pentose sugar constitute the nucleoside. Nucleoside, right. And nucleoside, nitrogen base plus pentose sugar by N-glycosidic bond. N-glycosidic bond unite the nitrogen base and pentose sugar, as well nucleoside plus phosphoric acid by ester bond. Right. Types of nucleotides as well nucleosides. No. DNA as well RNA, first one is adenylic acid. Adenylic acid. Guanylic acid. Thymidilic acid. Cytidilic acid. Cytidilic acid. These are the 4 types of nucleotides present in DNA. Then we add deoxy adenylic acid, deoxy guanylic acid, deoxy thymidilic acid, deoxycytidilic acid. These are the 4 types of nucleotides present in DNA, as well adenylic acid. Second one, guanylic acid. And third one, thymidilic acid. Not thymidilic acid, right. Cytidilic acid. And fourth one, uridylic acid, uridylic acid. These are the 4 types of nucleotides present in DNA as well RNA. Deoxyadenylic acid, deoxyguanylic acid, deoxythymidyllic acid, as well deoxycytilic acid. As well in RNA, adenylic acid, guanylic acid, cytidyllic acid and uridylic acid. These are the 4 types of nucleotides present in RNA, as well 4 types of nucleotides present in DNA, in which the common nucleotides are 1 adenylic acid and guanylic acid, as well cytidyllic acid. So which are present in DNA as well RNA. Present in DNA as well RNA. The special nucleotides are thymidyllic acid present in DNA only and uridylic acid present in RNA only. So the next nucleosides. Nucleosides, DNA as well RNA, deoxyadenosine, second one guanosine, deoxyguanosine, third one deoxythymidine, fourth one deoxycytidine. In RNA, adenosine, second one guanosine, third one thymidine, fourth one uridine. So these are the nucleosides in which nitrogen base plus sugar constitute the nucleotides. There are 4 types of nucleotides present in DNA as well 4 types of nucleotides present in RNA. Nitrogen base, sugar. By which bond? Nothing but N-glycosidic bond. N-glycosidic bond. So it is formed in between first carbon of sugar plus 9th nitrogen of purine. 9th nitrogen of purine. First carbon of sugar and 9th nitrogen of purine. N-glycosidic bond is there. As well, first carbon of sugar plus first nitrogen of first carbon of sugar as well first nitrogen of pyrimidine. So glycosidic bond is formed in between first carbon of sugar as well 9th nitrogen of purine, otherwise first carbon of sugar, first nitrogen of pyrimidine. Right, types of nucleotides. In nucleotide, the bond is nothing but nucleoside plus phosphoric acid. So it is formed by ester bond. Ester bond is formed in between, right? So once we observe adenine, it is purine bonded with thymine, sugar It is phosphate. So it is 5 prime. 5 prime means the open phosphate is present on a sugar, that is called 5 prime. Here open phosphate is there. It is totally one nucleotide. It is another nucleotide. It is another nucleotide. So it is phosphate. So here OH is there. It is 3 prime. Like the way it is thymine as well, it is adenine. Sugar as well phosphate. Sugar as well phosphate. Guanine, cytosine. Guanine, cytosine, sugar and phosphate, like the way it is the ester bond, OH is there, it is the 3 prime, ester bond, phosphate is, open phosphate is there, it is the 5 prime, it is the 3 prime. So these are the two strands. One strand polarity is 5 prime to 3 prime, the another strand polarity is 3 prime to 5 prime. So what is the 5 prime? 5 prime nothing but the open phosphate is present at the strand. The open phosphate is present at 5th carbon of sugar. It is the 5th carbon of sugar. So that is called 5 prime. 5th carbon of sugar, there is a phosphate, that is 5 prime. 3rd carbon of sugar, there is OH. The 3 OH is there. 3 phosphate is there. 3 phosphate show 5 prime. 3 OH show 3 prime as well. 3 phosphate is there, that is called 5 prime as well. 3 OH is there, that is 3 prime. So these are the hydrogen bonds. Here we observe every base pair. So this is totally constitute a base pair. Each base pair constitute of one purine, one pyrimidine, as well another base pair, one pyrimidine, one purine. Purine, pyrimidine. Always purines and pyrimidines are bound by hydrogen bonds to form a base pair. And a base pair consists of 2 nucleotides, one nucleotide and another nucleotide. What is the polarity of diester bond? What is diester bond? What is ester bond? What is diester bond? Diester bond for polarity. Diester bond means a phosphate. A phosphate consists of 2 bonds. So it is one bond, it is second bond. One bond, second bond. Right. It is the phosphate which form 2 ester bonds. So that is called diester bond. A phosphate having 2 ester bonds is called diester bond. What is the polarity of diester bond? Polarity of diester bond. Right. 3 prime to 5 prime. 3 prime to 5 prime. That is the polarity of phosphodiester bond. Polarity of phosphodiester bond, that is 3 prime. Nothing but 3 OH is there and 5 phosphate is there. 5 carbon have phosphate, 3 carbon have OH, 3 OH, 5 phosphate. In between these two, there is a phosphodiester bond. So the polarity of phosphodiester bond is 3 prime to 5 prime. Polarity of phosphodiester bond is 3 prime to 5 prime. It is very, very important. Diester bond polarity. Right. Ester bonds, diester bonds. Okay, dear students. So it is very important lesson, molecular basis of inheritance, especially the nucleic acids, especially the DNA. So we have the DNA related topics. The genetics is DNA related, as well biotechnology is DNA related, as well biomolecules are DNA related. So the structure of DNA as well RNA is very important for need purpose. It is better to understand the structure of DNA. Dear students, if you have got any doubts, please ask me. So I will clarify. So every base pair has 3 rings, already we said. So the purine consists of 3 rings and the pyrimidine consists of single ring. Totally 3 rings, so which maintain the 20 angstroms distance. 20 angstroms distance. Helic, length of helic, length of pitch. Length of pitch 34 angstroms, otherwise 3.4 nanometers between 2 base pairs is equal to 3.4 angstroms oblique 0.34 nanometers. 1 nanometer is equal to 10 angstroms, right? 1 nanometer is billionth of meter. 1 nanometer is equal to billionth of meter, as well 1 nanometer is equal to 10 angstroms. Right, the angle of helic, angle of pitch 360 degrees between 2 successive base pairs. Between 2 base pairs is equal to 36 degrees. So that is the angle of pitch. Angle of pitch is 360 degrees. Between 2 base pairs is 36 degrees. Okay, dear students. So the number is scrolling in YouTube. So please call the number scrolling in the screen.
ఎవరికైనా డౌట్ ఉంటే ఏమైనా క్వశ్చన్స్ ఉంటే స్క్రీన్ పైన స్క్రోల్ అవుతున్న నెంబర్కి కాల్ చేసి సార్ ఏమైనా క్వశ్చన్స్ అడగండి, సారు దానికి క్లారిఫై చేస్తారు.
So, dear students, the class is completed. Thank you.